So here we are going to discuss enteric bacteria again, and we'll discuss the ones that we did not discuss in the E. coli lecture. We're also going to put in here Pseudomonas as well. So as we discuss, our Enterobacteriaceae group of bacteria are going to be our facultative bacteria, meaning they can have both aerobic and anaerobic environments. And also they are going to be gram-negative rods. They are going to either have single rods or they can be diplo or in pairs. They're all going to be able to ferment glucose. They are all going to be oxidase negative. And they are going to have, which is called a peritrichous flagella. And this is basically on any part of their body, they can start to build flagella, and they can make many flagella. And this occurs when they want to be mobile. Now, we've already talked about some of these, so the major groups of this are going to be our coliform group, which are lactose positive groups. We discuss E. coli, for example, and then we also have our lactose negative, which is our PMP group. It stands for Proteus, Morganella, and Providentia groups, PMP. We also have our group that is not going to colonize us, and those are referred to as our pathogen groups, which we discussed already. Our Salmonella and Shigella are in these. And these are also going to be lactose negative. Now, many of these bacterium are going to be normal flora, and some of them are going to be primarily just gut pathogens, which is going to be our professional pathogen group. Now let's just talk briefly about Klebsiella pneumoniae. Klebsiella pneumonia is going to be a lactose positive. It's in that same group as our E. coli's with that respect. It has a large polysaccharide capsule, which is a major virulence factor. It's going to be able to be on our intestinal tract, respiratory tract. It's out in the environment. It's get in contact with it a lot. It's going to cause problems in people who are immune compromised, so nosocomial hospital acquired is very important, as well as in diabetics and alcoholics, people who have trouble with metabolism. 10% of the hospital infections are going to be associated with urinary tract infections, septicemia, postoperative wound, and endocarditis. Next up is our Serratus marcescens, and this little guy looks red because some of the more prolific in this group have this red pigment, which is proteus, and this is going to enable the bacterium to use or to make ATP much faster, and they can grow much faster. And so these again are going to be those facultative gram-negative rods. This is going to be referred to as a slow lactose positive. So it's still within that lactose positive group like E. coli and Klebsiella. And then it's going to be able to produce these red pigmented colonies. It is also going to be associated with UTIs, similar to our Klebsiella, wound infections, septicemia, and nosocomial infections. So people who are compromised are going to be more at risk with these pathogens. Now, an interesting note about Serratia is they were used back in the 1950s, 1960s. The military actually sprayed Serratia clouds into the Bay Area, And the purpose of this was to test whether or not the hospitals in the Bay Area were capable of mobilizing if there was a biologic threat. So there were, I believe, something like 11 cases reported, 3 hospitalizations and 1 death associated with this. But many hospitals thought that they saw an increase in cardiovascular disease, and when it was discovered that the military did this, it was believed that maybe that caused some of that increase as well. Now, another in this category is our Proteus species. This again falls into that whole facultative gram-negative rod. This one is lactose negative. So it cannot ferment lactose. It's going to be urea positive. So it can alter pH around itself. It has what we refer to as a swarming motility. And it also is going to be associated, similar to the predecessors, with UTI and wound infections. Now, another is Campylobacter Jejuni. So Campylobacter Jejuni is going to be referred to as an inflammatory syndrome pathogen. This little guy is going to cause enterocolitis. It causes about half the U.S. cases every year. In immune-compromised individuals, this enterocolitis will then progress to septicemia. It is going to be referred to as a microaerophilic. And that means that it likes air, but lower amounts of air than atmospheric air. It's also gram-negative, motile, curved rod. And so our next few are going to be curved rod, and so these are referred to as comma-shaped. Now, with Campylobacter jejuni, the reservoir is going to be poultry and wild birds. The major one is poultry for us, but also can be found in wild birds and other animals. The transmission is going to be not human to human, but ingestion of contaminated food. And it's thought to be present on about 47% of raw chicken breasts that have been tested. The diagnosis is going to be a stool culture. And then the treatment is going to be mostly supportive. So giving fluids. Some antimicrobials can be given if it's given earlier, and it will simply shorten the duration. Now, one of the complications with respect to Campylobacter jejuni infection is Guillain-Barré syndrome or Barré syndrome. This is an autoimmune disease, and this autoimmune disease is where you have your peripheral nerves are attacked. And so this is occurs in about 1 out of every 1,000 cases. And so it's thought to be about 40%, we'll call this GB, of GB syndrome is maybe triggered by our Campylobacter. Now we also have Vibrio Cholera. Vibrio Cholera is referred to as a non-inflammatory syndrome pathogen. It also is a gram-negative curved rod that's motile. So you can see the little flagella in these guys. Now the reservoir is going to be humans. And transmission is contaminated water in the developing world. And in the developed world, it's going to be more like shellfish, seafood. Now, the cholera has a lot of symptoms associated with it, and it can cause epidemics and pandemics. It's going to be associated with vomiting, severe watery diarrhea. And this is going to make these rice water stools. It also has dehydration. It can lead to, and so of course, as you know, water is very important, so it can lead to cardiac and renal failure, and death can occur within hours. Okay, so it has a 40% mortality rate without treatment. It produces, of course, its exotoxin. The exotoxin is famous, called cholera toxin. It is going to increase intracellular adenylate cyclase levels, which is going to cause an efflux of chloride. And this will ultimately lead to a massive secretion of water and electrolytes into the lumen. And this can be up to 2 liters per hour of fluid loss. And so, of course, so your AB toxin is going to be one of those where you have the active and then the binding component. And then the exotoxin is going to enter the cell by endocytosis, and then it's going to target the cell protein through ADP ribosylation. Now, worldwide, there are about 3 to 5 million cholera cases and over 100,000 deaths per year. And they're primarily going to be in developed countries. The treatment is going to be immediate replacement of fluid and electrolytes. So it's going to be supportive. Prevention is going to be public health, personal hygiene, proper cooking. There are a vaccine available. It's a 2-dose vaccine given in endemic regions, but it only lasts for about 6 months. And the vaccine is not available in the United States, and it's not recommended for most travelers. Recommendation is mostly prevention. All right, so next up is our Helicobacter pylori. Helicobacter pylori is another microaerophilic. And this is going to be a Vibrio shaped, so another kind of comma shaped. And of course our gram-negative rod, which is going to be very mobile. Vibrio cholera is going to cause gastritis. It's going to cause peptic ulcers, stomach cancer, it is not associated with diarrhea. So Helibacteria colonization is going to often occur without any disease. So likely many other contributing factors are necessary to actually cause disease by this bacterium. Now the reservoir is thought to be only human. And the transmission is going to be fecal-oral or oral-oral. The treatment is going to be with antibiotic and bismuth salts. And this, of course, will treat the bacterium, but not the ulcers. All right, and then we're going to let's talk about Pseudomonas aeruginosa. Pseudomonas aeruginosa is also a gram-negative rod, but it is not in this Enterobacteriaceae class. It's going to be lactose negative, oxidase positive, so it has some properties similar to what our staph had. It's going to have a motile polar flagella, which is different than our Enterobacteriaceae. So its flagella will be in a particular location. It also produces a water-soluble pigment, pyocyanin, and this creates what's referred to as a green pus, and it has a very fruity odor when it's grown in culture. So Pseudomonas has some virulence factor. It's going to have a capsule, which is its major virulence factor, and it's going to be able to form a slime layer. Alginate. It has an endotoxin because it is a gram-negative. So that is, of course, the LPS. It's going to have exotoxin A. And this is going to inhibit protein synthesis. And it's going to have proteases, so it can degrade our extracellular matrix proteins. So this is going to be our fibrin, elastin, collagen. And then it also has phospholipase C in its as part of its enzyme, so it can break apart lipids. Phospholipase. Okay, so Pseudomonas is going to be associated with many different types of diseases. It's going to be associated with corneal infections, and this is primarily for contact lens users. It's also going to be associated with external otitis, which is swimmer's ear. It is also going to be associated with hot tub folliculitis. Wound infections, especially burns. And it has about a 60% mortality in burn patients. Pneumonia. And so about 10% of nosocomial pneumonias. It's a major cause of pneumonia in cystic fibrosis patients, which is a leading cause of death of cystic fibrosis patients. And it also can cause septicemia with a 70% mortality rate. Now, Pseudomonas is going to be resistant to many antimicrobials. So it contains many plasmids and resistant factors. So it has a first line of antimicrobials. And that's going to be our ticarcillin or our piperacillin with an aminoglycoside. And then treatment should be tailored to the actual isolate the patient is infected with, and antibiotic resistance of that particular isolate has to be identified on a patient-by-patient basis.